So let's get into the nitty-gritty nuts and bolts of what we're actually going to be doing today. We're going to be talking about the structure and function. I believe we've already discussed this a little bit on 5-able, and so I'm not going to go heavy in depth on it, but I am going to still touch on it because I think it's so important for your success on the AP test. If you want to get that passing grade or even the 4 or the 5, you've got to understand the concept of structure to function. And it can seem very abstract, like your teacher just says structure and function, and you're like, So we're going to go over that a little bit, and then we're really going to get into the structure and the function of carbs, lipids, proteins, nucleic acids. So I have these gears up here, and the reason that I do that is because when you have something like a clock or a factory or whatever it is that you have built for your future engineer, future career, it's very important that all the pieces work together and that they work together at the same time in the proper way. You can also think of it in terms of the human body. Like, I have all these different organ systems. I have a nervous system, and a respiratory system, and a digestive system, and circulatory system. And if those systems don't work right, my body doesn't feel good. If you get ill with allergies, that's going to mess with your respiratory system, and your whole body can not feel great because of that. If you get ill with a digestive issue, your whole body does not feel great. If one thing is off, it's going to mess with everything else. So you want to have that proper structure so that everything can do its proper job, because they all are intertwined with one another. So let's talk about the individual ones and how they're intertwined. So kind of a repeat here. Structure, referring to the way something is built or arranged. So for example, the one that I always think of is my hands. These are my hands. If you think about the structure of my hand, I have a palm, I have a thumb, I have 4 fingers. The thumb is not a finger, just so you know. I also have a wrist, so I can twirl it around. And the structure of my hand is very important because it lets me do a lot of different functions. So it lets me hold a pen so I can write. It lets me text. It lets me take selfies. It lets me just hang out and click a mouse. I can use a calculator. I can put my hair in a ponytail if I want to. I can eat. I can do all these things because of my hand, and that's very important. Because let's say I didn't have a hand. Guys, I don't have a lot of stuffed animals. I don't have kids, so please don't laugh. This is just one of the few things in my house. But what if I was like this guy and I didn't have hands? And I just had a little nub. Just a little paw. So if my hand was like this, no fingers, I would not be able to text. I would not be able to write. Not with my hand the way it currently is. I would not have the same functions. And so that's why structure is so important, because different structure equals different function. Now, if I just had this, I guess I'd be really good at punching things, stamping things. But with this, I can do so much more. I've got fine motor movement and gross motor movement. That's small and large motor movement, or the ability to move your hands for any of those who want to be doctors or physical therapists someday, just so you know. So, all right, let's get into our first topic. So we're looking at carbohydrates. Carbohydrates. You may sometimes hear expressions like, I don't eat carbs, or I love carbs. Whenever people talk about carbs, they're talking about certain foods. What kind of foods do we typically find carbs in? Can you guys type that into the chat box? Where do we typically find carbs, carbohydrates, in what foods? I'll give you a hint. There's a version of it on the page. Sugar! Yes, we love, I love sugar. So your breads, pastas, starchy vegetables, sodas, all that stuff. It's gonna, potatoes, yes, rice, those are good, very good. And there's gonna be different types of sugars, different types of carbs, but typically they're gonna be white or brown if it's food that we eat, as opposed to, say, grass that cows eat, which is a carbohydrate, but it's one we actually can't process. But so carbohydrates are our body's main energy source. If you're thinking of a metaphor for this, you would be thinking of gas in a car. If you want the car to go, you need to put gas in it. If you want your body to work, you need to put carbs in it. That's your basic energy source. And the reason it is because its structure can be extremely simple. It can be very easy. So there's going to be two major types. That simple sugar that is really easy to digest. You're like, all right, I got to run a marathon. I need energy now. And so someone hands you some spaghetti and woohoo, go to town, because you've got all those carbs from that pasta. But then on the other side, you can also have something called starches. And starches are your more complex carbs. And while they're still an energy source, because of their structure, they release much more slowly. So you don't get that energy quite as quickly. So typically, again, your white foods are going to be those simple sugars. So like pure sugar, it's white. That's your simple sugar. That's the easy, fast energy. Whereas your darker colored foods like potato skins, not the potato itself, but the skins, brown rice, whole wheat pasta, those are going to be more starchy, more whole sugar. That's going to take longer to break down. And so it's going to have a bit of a different function. So when we talk about this, we tend to, let me see, focus on the main monomer. Monomer. If you don't have the word monomer in your head, and if you don't have it on a flashcard somewhere, I strongly recommend you put it on there, because if you say it in class, your teacher's going to be like, that kid is so smart and I'm so proud of them. And you're going to be like, yeah, I am. I studied. I went on Fiveable. So do that. But the monomer is like the basic building block. So if you think of, let's say, house. So you've got a house. It's very cute. You're financially stable. You're in your late 20s. You can afford this house because you have a good job, because you got good grades in college and you have no debt. It's great. You have your house. What are houses built out of? What do we build houses out of? Your dream house. What would you build it out of? Let's say that this house is made out of bricks. We're going to go with something super simple. So I've got my bricks. Look, I'm an artist. That's a brick. Say it's a brick. The brick is my monomer. It's the basic building block of my house. If I put a bunch of these bricks together, I get a house. So we have those with biomolecules as well. They have a bunch of itty bitty pieces, and we put them together, and then we have a big something, a macromolecule, if you will, a big molecule. You can also think of this in terms of chemistry. You typically start out with atoms, and you put the atoms together to make molecules. Same kind of concept. You have a basic teeny tiny subunit, and you build it up into something bigger and bigger. In this case, we've already got the molecule. We have this thing right here. This is our monomer. It's called a monosaccharide. And the monosaccharide, mono means one, saccharide, like saccharin, means sugar. So it's a single sugar. That's our building block that's going to build all the rest of the sugar. So when we put these guys, our monomers, together into groups, we can then stretch them out into these chains. Or in some cases, they're linked. But in this case, we have a chain of monosaccharides. And those monosaccharides chained together are going to provide energy. Where do we store the energy in these monosaccharides? Can anyone tell me where the energy is stored? Where do we store energy? You may know this from chemistry as well. The bonds, yes, Salika, thank you. The bonds are where we store our energy. So any bond, you are releasing a little bit of energy. Right here, I've got these 3 sugars. That means I have 2 bonds. That means I have 2 bonds of energy worth that I can access and I can utilize. And so, yay, because we love energy, because we like to stand up and walk around and dance and party and all these things. And to do that, we need energy. And that energy is stored here in the bonds. So now this would be a very simple sugar. I know it says starch overhead. We're going to get to the starch down here in a sec. But this is a pretty simple sugar to begin because it's just the 3 glucoses lined up together. And all you have to do is chop one off, there's your energy. Chop another one off, there's your energy. It's not very difficult to get that energy. And so this is important because that single bond structure is what holds the energy. And all you have to do to get that energy is break it down. So you think of like, you got a snack, you've got your popcorn. I eat my popcorn in front of you guys. Sorry, I hope you have snacks too. I eat my popcorn, it goes down, I digest it. What happens to those bonds when they're getting digested in my stomach? What happens to bonds when I start digesting them? What happens to those bonds when I start digesting them? They break down. Very good, Sandra. So they're going to break down. So I just ate that popcorn, a carbohydrate. It's in my belly now. And the acids and enzymes and things like that are going to break it down. And they are specifically going to break down all these bonds. And that's going to release energy that I can then use to keep putting on this show. So that's a good thing. That's good structure that I can access by having this monosaccharide, this 6-carbon sugar. But now here's where we get to the more complex type of sugar. So we have these simple ones. You can have glucose, which you find in your blood. That's when, especially if you're ever diabetic, if you know someone who's diabetic, they have to do the finger prick pretty regularly. And they do that to test their blood sugar. They're testing their glucose. Whenever you get a physical, they'll often start checking for those as well as you get older. We can also have galactose, which is from milk and ice cream and all the other good stuff. And then we have fructose, which is typically coming from plants. So whenever you eat apples, strawberries, blueberries, even vegetables, that's going to be fructose. And I know this one's kind of blurry, but notice how they're all rings, whether it's a hexagon like my glucose or a bigger hexagon or even a pentagon. They're always in this ring of some sort. The ring may be a triangle, it may be star-shaped, but it's in a ring. And then we join all those rings together to make the polymer. And that polymer, like when we have 3 of these together, poly meaning many, saccharide meaning sugar, many sugars, takes different forms depending on which monosaccharide you use. So in this case, we've got something called starch. And this is a pretty good polysaccharide because it's fast energy, but not quite as fast. And so it sits in your belly and it takes longer for your body to digest it. Just looking at this image of starch and all these cute little branches, can anyone think of why starch might take longer for you to digest and access that energy? Why does starch take longer to digest? Why does starch take longer to break down? There are more bonds in starch. Very good, Sandra. So if you look at this starch, it's not just one straight pretty line. That's great for like Kool-Aid and soda and candy. It's really, and that's why it tastes so rich and sweet to us, is because it's those easy bonds breaking. But starches, you may notice when you taste them, they don't taste particularly sweet. Like you don't go to get a piece of bread when you have a sugar fix or you need to get one. You don't go for bread, you go for like candy or fruit if you're good. Starches though, they're not that straight, perfect, just line. It's gonna be like this, where everything is bent and branched. And that is because they usually have double bonds. Now, if you're trying to break down energy, which one is gonna be tougher to get energy out of? Breaking a single bond or breaking a double bond? Which one's gonna be easier to get energy? Double, very good. So double's gonna be tougher. You've got to break it, and you've got to break the double bond. And that's why whenever you eat starches, those starches take longer to digest. That's why they tell you, oh, don't eat simple sugars. Don't drink the soda. Have a nice starch, a nice complex carbohydrate. They use complex because it is more complex. It's not just in this straight line. It's going to be with those rings and branches. So that's why they prefer them. At least your medical doctor normally does. They say, eat your complex carbs, no simple sugars. And then you say, you only live once, gonna eat the candy. At least that's what I do. So I'm sorry if I'm a bad influence, guys. So again, going back to that, we can have the simple ones, our sodas, candies, milks, chocolates, things like that. Typically, because these break down so fast, that's why you get this blood sugar spike where you're like, we have all this energy. Maybe you felt this when it's late at night and you're studying and you've got to stay up for at least another hour. But then you hit the top and you go whoosh, and your energy crashes. And next thing you know, you're waking up with your head on your desk because you fell asleep at your desk. That's a sugar rush. And that happens because you break down all that sugar so fast, and then there's nothing left. Your tank is empty. You are a car on the highway with no gas, and you are stuck there unless you get more sugar. So I guess you could always go eat more, but it just gets worse and worse as it goes on. Whereas if you eat a more complex carbohydrate, like brown bread, brown rice, things that are not white when you bake them, chocolate doesn't count. You'll notice the blood sugar doesn't spike as high, and so you slowly access the energy because it takes longer to break those double bonds, but you also slowly lose energy as opposed to crashing down. And so a lot of the times, especially with athletes, they say complex carbs, otherwise you're gonna go play your basketball game and you're gonna run out of energy like 2 minutes into it. Y'all ready to talk about some lipids? I love lipids, I love fats. They are delicious and they're good for you. There used to be a big trend where they would say, don't eat fats, they're not good for you, but they are extremely good for you and they make up a big majority of your body, and they are supposed to. So lipids, one of the most important things you have to know about them, they have what's called a non-polar structure. Non-polar. So if something is non-polar, is it gonna be hydrophobic or hydrophilic? If it is non-polar, is it hydrophobic or hydrophilic? Non-polar. Hydrophobic or hydrophilic? Hydrophobic. Very good, Salika. So fats are going to be hydrophobic. So if you're ever, here's the best metaphor I have. If you're ever one of the lucky ducks who got to wash dishes after Sunday morning breakfast and you drew the short straw, you got to do dishes. And if you're like me, you don't do it immediately because you're a procrastinator, and that's what I do. And so, but then when you come back to wash the plate and someone had fried eggs with the runny yolk, if you ever tried to wash yellow egg yolk off a plate, you have to like scrape it and it won't come off just with water. That's because egg yolk is the fatty part of the egg. That's the long-term energy that the baby chicken inside the egg is living off of. The white is the protein. That's why you always hear the stereotype, I will have an egg white omelet, because they just want the protein without the fat. The whites are the protein, we'll get to that. The yolks are fatty, but they're non-polar. So when you've got them on a plate and you're running water over them, you can't soak off egg yolk. It doesn't work that way. You have to actually brush it off with soap because it's non-polar. It is hydrophobic. Same thing goes for my best friend, the avocado. I love avocados. And avocados are a great source of healthy fat, but they're non-polar. Once I let that dried avocado stick to my plate, it is stuck there for as long as it wants to be. So not great to clean up, but lipids are really good for you. When you have certain types, there are some that are obviously not good, but anything out of control is not good for you. I want to make that clear. But so lipids are going to be nonpolar, which is critical to their structure, which is critical to their function. They're a secondary energy source. So when you are on that sugar high and you've run out of carbs, your body will start burning the fat that you have eaten or that you have stored. It can also operate as chemical messengers. So all of your hormones are going to be based in lipids because they're nonpolar. They don't dissolve in water. It can be insulation. So when you like go outside and it's cold, it doesn't murder you. And finally, they're major components of cell membranes. So we're going to get to that in just a second. Actually, I'm going to do it right here because I have the picture right here. What's this thingy right up here that I'm circling? What's that guy? Phospholipid. Very good. So this is a phospholipid. Phospho, as in it's got phosphorus, so you can see that phosphate up here. Lipid, as in it's got these lipid tails, more to these. But because it's got this hydrophobic tail, it's scared of water, it actually becomes waterproofing because you can put these guys together and make something that is hydrophilic. It likes the water versus something that is hydrophobic, and you can protect them by stacking them on each other like so. You've probably seen this before because you go to school. But so they'll stack these guys together to protect the hydrophobic tails and then have the hydrophilic heads that can interact. It's a good structure because cells are naturally going to interact with water. We want them to, otherwise we would dehydrate and die. And it's also a good function because then let's say I've got some food, I need to get into my cells, it can still find a way to pass through. So go us, we've developed well. So let's get into this lipid structure. Again, we're going to have another monomer. Monomer, get that word in your brain. But these are going to be a little bit different in that they're just going to have a glycerol. So that's this long chain here. And then 3 fatty acids. 3 fatty acids, 1 glycerol. And that's going to come together to make a triglyceride. Tri as in 3, glyceride as in glycerol. And so this is going to be your typical fat structure. Obviously, there are differences, like up here where we have the phospholipid, we've only got 2 fatty acid tails and we've got a phosphate. Whereas here we've got the 3 fatty acid tails and a glycerol, but different structures are going to give them different functions. So when we've got this triglyceride model, this is typically what we're going to store somewhere on our body. Like my snowman here, he's storing some stuffing, but that's where you would store most of your long-term energy. Whereas a phospholipid, I'm not looking to store energy. That is not its function. That structure is waterproofing. So with that structure of keeping the water out, it provides the function of waterproofing. These guys, on the other hand, their structure allows me to stack. Because on here, it looks like I've only got 1, 2, 3, 4, 5, 6 bonds. And remember, those bonds are where we store the energy. But then as we break them, we use the energy. But these don't have to be 6. Those tails can go on and on and on and on and on. So how does the structure impact their function? So kind of like here, we've got a long chain and we can chop off carbon hydrogens in order to get energy as we need. But here we've got our double bond. And that double bond, is that easier or harder to break? Is it easier or harder to break a double bond? Harder, very good. So unsaturated fats, those are the fats that are harder to break down. And so they really get stuck there. They are long-term energy storage. Whereas these, the saturated fats, they're also energy storage, but they're easier to break down and it's easier to get rid of them. You can tell an unsaturated fat because it's going to have these bends every time there's a double bond, just like the starch. It's going to bend and twist and bend and twist. Let's talk about some protein. Yes, that's spinach. Spinach is very high in protein. That's why Popeye eats it and he gets gains. And that's a good thing because he's always fighting for the lady. And apparently he needs muscles to do that. So 1950s cartoons, that's what they say. So proteins are going to have a ton of functions. And whenever your teacher asks you, what is the function of a protein? And you stare back like a deer in headlights and you're like, it does work. For me, that's pretty adequate because long story short, they do a lot of work. They do work in muscles, so they contract the muscles so you can pick up heavy things and put them back down. They are involved in hormones. They are involved in protecting your body. That's a job. They are involved in transport, moving things through cells, all of these things. And their structure is so highly specialized. Like if you thought there was a lot going on with carbohydrates and lipids, let me blow your mind. So the monomer of a protein is called an amino acid. So we've got it here. It's got a nitrogen. It's got a carboxyl group over here. And it's also got this thing here. It's got an R group. The best way I can describe R, I think it stands for remainder. So remainder group. Think of in math whenever your teacher asks you to find X and Y. X and Y are the variables. You can replace them with literally anything. R is like our science version of X and Y. With this R, it has the potential to grow up into anything. It can be any protein. And so depending on what different R group we put right here, that's the different amino acid we have. And depending on the different amino acid, that's the different function. So let's look at that. Okay, this slide's going to take a minute, and your brain may say, ouch, in a second, but follow through. I promise it's not that bad. So proteins are literally just a single long string. And I know this is a measuring tape, follow my metaphor. Each piece is a single amino acid. So I've got my single amino acids. Now, I want you to think, you keep protein in your cells in your body, right? And they come in these long strings. You ever put your headphones in your pocket and then taking them back out and they're this crumpled mess? Protein does the same thing when you try to pack it in a cell. So it doesn't stay in this nice string. It just gets tangled up. But keep in mind, I told you about these R groups. Those R groups, depending on what they are and their polarity and their charge, are going to bond with other parts of the protein. And so it's going to fold in a highly, highly, highly specific manner. And so it folds up, and at first it'll start really simple. It'll be this nice, pretty folding. And you'll be like, oh, that's so organized. That's such a good aesthetic. That's wonderful. Yes. But then there will be another bond that'll be exposed, maybe this silver piece down here. And it's going to say, I must bond with this amino acid. And so it goes, whoosh. So it folds, and then another piece has to fold, and then another piece has to fold. It looks like a tangled lump by the time it's done. You can see it down here on quaternary structure. But that's a good thing. Because if you've studied proteins and you've studied enzymes before, they always have a substrate, and that substrate has to be a perfect shape that fits with your protein. And if it doesn't fit, it doesn't work. And if you, for example, have a non-functional lactase protein, you can't digest lactose and you can't eat ice cream. I apologize to anyone who may be in the audience who cannot eat ice cream, sorry. And that's all because you didn't fold your protein right. Imagine if every time you didn't fold your laundry right, you couldn't wear it. Every time you don't fold your protein right, you can't use it. It's highly, highly, highly important to the function. Structure and function is most applicable to proteins. If the structure of the protein isn't perfect, there is no function. All right, last one. And this one I love because I get to go on and on about superheroes. So nucleic acids are DNA and RNA. Those are the big ones that we always talk about with them. So I don't know if you guys are familiar with these television masterpieces of superhero characters, but what do they have in common? Maybe if you're one of these people in their origin stories, how did they become superheroes? Something happened to them. Something inside them. I'll give you a hint, it has to do with nucleic acids. What happened to these two particular superheroes? Okay, if you haven't seen them, Google them. Spider-Man, Danny Phantom. Both of them became superheroes because there was a change in their DNA. Danny Phantom stumbled into the ghost portal. Yes, very good. DNA mutation. Danny Phantom stumbled into this little ghost portal. And if you remember the classic theme song, his molecules got all rearranged. His DNA was changed, and therefore he was changed. He was a ghosty. Spider-Man, bit by a spider, changed his DNA into part spider DNA, and then he could like swing around and shoot webs and all sorts of things. Now that being said, fictional stories. I'm not telling you to go get bit by a spider or to wander into a ghost portal. You're not going to get ghost powers. You're not going to turn into Spider-Man. I don't mean to crush your dreams. It's just, those are kind of science fiction. But nucleic acid, depending on their structure, determines your function. I need my DNA to have a certain structure so that I can do certain stuff and so I can look a certain way and so I can have certain skills. Danny Phantom and Spider-Man, their DNA got changed and it literally changed them into a ghost and a half spider dude. Structure affects function. So when we talk about nucleic acids, we're going to talk about two particular monomers. Those monomers are going to involve ribose and deoxyribose. What's the difference between ribose and deoxyribose? How do we use those differently? All right. So ribose is going to be for RNA, ribonucleic acid. Deoxyribose is going to be for DNA, deoxyribonucleic acid. And so these are going to have very, very, very specific functions. We won't get into that because that's a little bit later unit and we're kind of running out of time. But they all have this specific function of having a phosphate and a 5-carbon sugar and a nitrogenous base. And these are going to link together into these cute little DNA double helixes. Hopefully you've seen some documentary about Watchmen and Crick. We know that make the DNA and it bridges together, la la la. And the sequence of that DNA determines your function. Do you have spider powers? Do you have ghost powers? If you don't, it's because your DNA does not have the proper structure. So you're going to get into this later, really later, depending on your teacher. But DNA is really important to making protein. So we just talked about protein, but to make protein, you have to have proper DNA. So this comes back again to that proper structure and function. Your DNA has to be in that perfect sequence that makes you, you. If it's mutated to give you spider powers or ghost powers or whatever, then it's not going to do the same thing. It's not going to create the same proteins. When Danny Phantom's DNA got changed, he made different proteins, and those proteins are what made him go ghost or whatever. So his structure changed, his function changed. He didn't have this same DNA double helix. He still had the double helix, but the order of the molecules, the nucleotides, was completely changed. So this is something we're kind of wrapping up here that I give all my kids. And I really hope you take this and use this. But this is one of the best ways that I learned to study in college. And I didn't learn this until college. But I think it's really cool because it's a way to organize your thoughts. And I'm sure your teachers have given you like graphic organizers before, but I love doing this with science when I have to compare multiple things like the 4 biomolecules, because I can add as many columns and rows as I want for all the different characteristics. But the stuff we covered today, we covered the structure of monomers, so monosaccharides, glycerols and fatty acids, nucleotides and amino acids. We covered the structure of the polymers. So for carbohydrates, that polysaccharide, that starch. For lipids, you could say triglyceride, but they're pretty monomer based. For nucleic acids, the DNA and RNA would be your polymer. And for proteins, when I get my big long chain of amino acids and I crumple it up, that's my polypeptide, many peptides, many proteins. And then I went ahead and added on the function here. So I can study this. I can erase everything and fill in the blank. It's just another way to do flashcards, but have it all in one place. So